I want to thank Dr. Sayer for inviting me. It's a great pleasure to be here. This is my first opportunity to come anywhere in Scandinavia, so this is a big thing, and I've been enjoying the city here today. So let me start by so I'll be talking about this long title here about the ONOO-Cycl that I think many of you know something about, and I'm going to start talking about it in the context of heart failure, which is perhaps a big surprise to you. I published a big paper on heart failure as an ONOO-Cycl disease late last year, and I think for the first time we have extraordinary evidence that the cycle is causing a specific disease here. And so I'll be talking about MECFS, MCS, fibromyalgia, very little actually, there's almost nothing on that. I'll say something about electromagnetic hypersensitivities. And basically the question is, what is the evidence and how should we treat these diseases? You know, that's the central question here. So a lot of this talk comes from 6 of my publications, which are listed here. And what you'll see here is, first of all, my 2009 big review on multiple chemical sensitivity, which is a very important paper. And up until recently, probably the most important paper I've published on anything. My book in 2007, some of you know, and then recently these 4 papers, and each of them has important take-home lessons, and so I'll be talking about those quite a bit. So now, as I think many of you know, what's called the ONOO-Cycl originally came from 2 different sources. One is a series of very well-documented biochemical and physiological mechanisms. So there's nothing new about those. It's just simply you put them all together and you come up with this very complex cycle. And secondly, the properties of the related conditions, and I refer to them as diseases because I believe these are true diseases. MECFS, multiple chemical sensitivity, MCS, fibromyalgia, post-traumatic stress disorder, is also discussed in some detail in my book. Now, whenever you have a scientific theory and you're trying to test that theory, you always want to test it based on data that's totally independent of the data that was used to originally propose the theory, okay? That's very important, right? Because obviously the theory is aimed at explaining the data you already have, and then the question is, when you get a lot of new data, does that confirm it or not? And so basically what I'm going to do is talk about heart failure in that context, because I think heart failure is the fit is absolutely stunning, and I think you'll be more or less convinced of that when we're done with this. Although I really suggest you read the paper, because it's a public access paper, so anybody can access it for free. Now, so basically what I will be discussing, so this is an outline basically of my talk. First of all, the cycle and how it can be tested as an etiologic mechanism for a specific disease. And then I'm going to talk about heart failure as a extraordinarily well-documented example of an ONOO-Cycl disease. And I'm going to talk about evidence with MECFS, particularly some new evidence which has been accumulating over the years that was not available when this theory was originally proposed. And then I'm going to outline the evidence on MCS. That includes some recent evidence, but most of it is in my 2009 review. And I'm going to discuss the Hetherington and Battershill paper that was published in early last year, that purports to review the toxicological evidence regarding what they call idiopathic environmental intolerance. And as some of you may know, they conclude there isn't any. And as you might guess, I'm going to come to a very different conclusion here. So I think it's interesting to go through the discussion of their paper, and you'll see some of the things that are there and also some of the things that are not there. Then I'm going to discuss electromagnetic hypersensitivity and why we think that the mechanism of sensitivity is very similar but not identical to MCS. Okay, so and finally we'll talk about some things regarding therapy, and my discussion of therapy will be, I mean, there are lots of ways of trying to treat these, I think, and it's certainly, my discussion here is not going to be complete in any sense. So the etiologic theory I'll be discussing here is focused on nitric oxide, which reacts with superoxide here to form an oxidant product, peroxynitrite. And peroxynitrite is the most central thing in the whole ONOO-Cycl, as you'll see shortly. And this is the cycle, and for those of you who haven't seen it, you will gasp and say, hey, how can I possibly understand this? It looks like gobbledygook. And what I'm hoping is that by the time we're through, you'll feel a lot more comfortable with it. And so what I'm going to do here is just talk about what these different elements of the cycle are, and a little bit about how they fit together. And then we're going to talk about a lot more about how they fit together to form complex vicious cycles. Okay, so here's nitric oxide, NO, reacts with superoxide over here to form peroxynitrite, ONOO-. So that's the reaction we just discussed. And peroxynitrite, being a potent oxidant, produces oxidative stress, which is an imbalance between oxidants and antioxidants. So you got too many oxidants and not enough antioxidants. And both peroxynitrite itself and the oxidants involved here in oxidative stress are capable of activating NF-kappa B, which is a transcription factor, which activates a lot of different genes and has a central role in essentially every inflammatory condition in existence. Okay, so this is very important in terms of both chronic and acute inflammation. And NF-kappa B, as I said, is a transcription factor. So it goes into the nucleus and it can act on various genes. And among the genes that are activated by it is the INOS gene, the inducible nitric oxide synthase. So this is an enzyme which normally is present in very low levels in cells, but under inflammatory conditions can go way, way up and therefore produce a lot of nitric oxide. And it turns out that NF-kappa B can also activate these 5 inflammatory cytokines. And some of those cytokines can also act independently to activate INOS, and some of them can actually go back and activate NF-kappa B. So this is a double-headed arrow. So some of these guys can come back and activate NF-kappa B, so they can go there and come back and activate INOS. And then you get a lot of nitric oxide. So we have here, you can see a cycle, complex cycle, and I'll talk about it a little bit more in a minute or so. But before going into that, I want to talk about some of the other elements of the cycle, okay? And so one of the things that happens is that there's a very complex cascade of events which can occur from nitric oxide and from peroxynitrite, which leads to mitochondrial dysfunction and lowered ATP production, okay? And And one of the things actually is quite interesting about heart failure is that almost all of this cascade of events, which I postulated a long time ago, have been shown to occur in heart failure. So we know that these things really do work in real diseases, so you get a depletion of ATP, and that has a number of effects. One of the effects is that it activates via two mechanisms these receptors known as NMDA receptors. These are receptors for glutamate, and when they're activated, they allow calcium to flow into the cell, and so you get excess intracellular calcium. And what excess calcium does is it activates a couple of other nitric oxide synthases. Okay, so these are nNOS and eNOS are both nitric oxide synthases that are calcium dependent. So they're essentially completely inactive in the absence of elevated intracellular calcium. But when you get elevated calcium, and there are a number of ways you can get this, this activates these two nitric oxide synthases, and they of course make more nitric oxide. So you get nitric oxide from these guys and from that one, and of course that can then go in and do all these other things. Okay, so basically so I've talked about all of the elements of the cycle except for two of them. Surprise, surprise. One is that one of those two are these TRP receptors, and these are also receptors that are capable of allowing calcium to flow in the cell. So when they're activated, they allow calcium to flow in the cell. They also can act, and at least for two of them, and probably for several others, they can also activate the NMDA receptors. So that's where these arrows come from. And I'll talk about some diseases where this is where these are elevated, but there are also some where we don't know, and one of those is MECFS. This is the only thing in the cycle we don't know about with MECFS. Everything else we do know. And the other thing which happens is over here. This is BH4. This is what's called tetrahydrobiopterin. Okay, that's a mouthful. And it's a cofactor for a number of different enzymes. And among the enzymes that it's a cofactor for are these three nitric oxide synthases. And so what we know is true, and we actually did some studies on these arrows in one of the papers that I showed you before. We know that peroxynitrite can oxidize BH4, okay, and cause a depletion in it. And when it does that, what I argued was that you actually get an increase in peroxynitrite. And I don't think I'm going to try to explain all that right now, but it's true. And we've shown that that's true. Okay, so basically you have another cycle over here. Now, so basically, you know, when you look at all these arrows, it's really almost impossible to keep track of them, right? And so what I've done is to divide that cycle up into different parts by using colored dashed arrows. Okay, and this is the inflammatory part of the cycle over here, and this is the part that we first started talking about. Nitric oxide superoxide forms peroxynitrite, which causes oxidative stress, turns on NF-kappa B, turns on the cytokines, turns on eNOS, gives you more nitric oxide. And so what you can see here, you already have this cycle, you have that cycle, you have this cycle, you have that cycle. You already have multiple cycles. Once they start going, they're going to tend to go on their own, okay? And that's the crucial point. If you have a vicious cycle, once it gets turned on, it tends to propagate itself over time, okay? But that's only one part of the overall ONOO-Cycle. Okay, so we already have multiple cycles, and then we've got this cycle. This is the mitochondrial dysfunction leading to lowered ATP can activate the NMDA receptors in two different ways, and it then produces increased intracellular calcium, which turns on nNOS, which gives you nitric oxide and turns on this mitochondrial cascade. Okay, so this is all another vicious cycle, complex vicious cycle, right? Once it gets going, it's going to tend to go on its own. Let me just say, again, I mentioned this before, all the mechanisms on this are well-described mechanisms. They're not things that I made up, okay? But basically you put them all together and you get these cycles, and that's, so that's another cycle, okay? So here's a third cycle. This is the one we just discussed before, where peroxynitrite oxidizes BH4, you get a depletion of BH4, and this is then thought to increase peroxynitrite. And so you have a small cycle there. And then there's another cycle going through some of the TRP receptors. So some of the TRP receptors we know are susceptible to oxidants. So they can get activated by oxidants, by superoxide, by oxidative stress oxidants, and so they can then act in this way to produce another cycle. Okay, so you get oxidative stress, and then it goes back up to TRP and intracellular calcium, and so forth. So here's another cycle. Once it gets turned on, it's going to tend to propagate itself over time. And this is a fifth cycle. I got to keep my numbers straight. And the central actor in this cycle is an enzyme called the calcium ATPase, which is present in the plasma membrane of cells. And its role is to keep intracellular calcium levels low, okay? So what it does is when calcium accumulates in the cell, it pumps the calcium out and lowers intracellular calcium. So that's an important thing. And so basically when that activity is low, of course, you end up with too much intracellular calcium, right? And it turns out that activity is inactivated both by oxidants and specifically by nitration, by peroxynitrite. So there are multiple effects by oxidants on that calcium ATPase. And so one of these arrows is a mechanism for peroxynitrite inactivating the calcium ATPase, and the other one is for other oxidants inactivating it by a different mechanism. So these are different mechanisms, they're not the same. And but it's also true, of course, calcium ATPase requires ATP. So whenever you have mitochondrial dysfunction, you're going to have a lowering of the calcium ATPase, right? And so this mitochondrial cascade, which is indicated here, also has a role. And when you see a depletion in ATP, of course, it will, in effect, raise intracellular calcium by lowering the calcium ATPase, okay? And let me just tell you, it's kind of interesting, when I studied the heart failure situation. In heart failure, there's another calcium ATPase that pumps calcium into the sarcoplasmic reticulum. Rather than pumping it out of the cell, it pumps it in the sarcoplasmic reticulum, and that, it turns out, in the cardiac cardiac cells is more important, and that also has similar properties to the plasma membrane calcium ATPase. It is inactivated by peroxynitrite, and it is inactivated by oxidants in other ways. So sometimes you see variations on a theme, and that's one of the things that I think one often sees. So basically, and I'm just going to go through these quickly, if you look at these, what do you have? You've got all of these things, and each one is amplifying the other, right, through their common elements. So this one's going to amplify that one, which is going to amplify that one. This is all through peroxynitrite now, and this is the only common element between this one and the others is peroxynitrite. And then it's going to amplify that one. And so all of these things, and then finally this one. Each of these are going to amplify the others. And so what I think is true is that the challenge in therapy is how do you downregulate this whole massive mechanism, okay? That's the crucial problem in therapy. And that's the thing that I think that, obviously medical people have to deal with, and people who suffer from these diseases have to deal with. So that's the if there's a simple take-home lesson there, it's obviously not very simple because you have to kind of see these various cycles. But if there's a simple take-home lesson, that is it. You have to downregulate these cycles in order to treat these diseases effectively, and so that's the challenge. Okay, now there are what I call 5 principles that underlie the cycle, and these 5 principles can be used then to test whether a particular disease is apparently caused by the cycle or not. Okay, so these are all ways of testing whether a particular disease may be caused by the ONOO-Cycl. So the first principle is that cases of the disease can be initiated by short-term stressors that increase cycle elements. So the only way in which you can turn the cycle on is to raise cycle elements, okay? And it follows from that any kind of short-term stressor that initiates cases of the disease must be able to raise cycle elements. And so that's testable, and we'll talk about how some of these things work. Okay, so that's the first principle that's testable. Second one is that in the chronic phase of illness, if it's produced by the cycle, then it follows that the cycle elements will be elevated. Okay, and so that's testable. The symptoms and signs of illness must be generated by one or more elements of the cycle. Okay, so if the cycle is causal, it has to be able to cause whatever correlates there are for the disease, and so that's testable, and so that's a third thing. Now the fourth principle is something that's probably not obvious to you at this point, but that it is that the basic mechanism of the cycle is local. And so because it's local, it can be localized in different tissues in different individuals, causing a variety of different diseases, okay? And the reason that this is primarily local in nature is that the 3 compounds involved, nitric oxide, superoxide, and peroxynitrite, all have very short half-lives in biological tissues. So they don't go very far from where they're made to where they're destroyed. But in addition, the mechanisms of the cycle, all those arrows, we haven't actually talked about what they are yet. Well, I guess we've talked about a few of them. Act at the level of individual cells. So we're not talking about things that are, that influence the whole body equally. We're talking about things that, because they involve local mechanisms and because these 3 compounds can be localized in specific tissues, and when they are localized, they don't go very far outside of that to impact other tissues, that you have a primary local mechanism, okay? And so basically what I argue is that this allows for great variations in tissue distribution from one patient to another, producing a huge spectrum of illness. And now, the point here is not that there are no systemic changes. I'm not arguing there are no systemic changes. Clearly, when you have oxidative stress, you see a depletion of antioxidants to some extent systemically, okay? And when you have inflammatory cytokines, you see those to some extent systemically. In fact, I mean, those are typically things that people measure systemically, not in the specific tissue. And it's also true, for instance, with BH4 depletion, that is to some extent systemic, okay? So you can see it by taking a blood sample, regardless of where, what the specific tissue is that's impacted. So there are some systemic things, but the point is that we think that there are one or perhaps several tissues that are impacted in each of these diseases that are then responsible for producing the characteristic symptom of the diseases, and so that's a key factor. So the question is, is the mechanism of the disease primarily local? And if it is, then of course it's another type of evidence supporting the ONOO-Cycl as a mechanism here. Okay, now, but one of the things I think that is true, and I'll just, with these diseases, MECFS, with MCS, with fibromyalgia, and so forth, it's very common for patients to come in and have this whole spectrum of complex things going on. And it's very common for the physician to throw his hands up and say, I don't understand this, it must be psychiatric. And of course, that's totally illogical, right? But it happens, and I'm sure some of you know it better than I do. So, so it is the case that, if you have one tissue that's impacted, there may be a higher chance that other tissues will be impacted as well. You'll have, in effect, multiple diseases occurring, either because the person might be genetically susceptible or because there are, to some extent, systemic effects that might tend to make other tissues more susceptible, and or both. And so it's not uncommon that you have this complex spectrum that doesn't fit the sort of, okay, if a patient has a disease, it should be fit into some specific category, and therefore, we do the usual differential diagnosis and say, okay, that's the disease, that's what we have to treat. In fact, it's very common that these are quite common. Did you have a question? No, okay. This is what you expect from the mechanism, okay, that a lot of these are going to be complex. And so I think, there is a conflict between this and conventional medicine. I discussed that in my book in a whole chapter, that there is a conflict here. And but the fact is that traditional medicine with specific diagnoses doesn't work very well in a lot of areas of medicine, and this is at least a partial explanation for why it doesn't. Okay, now the fifth principle, and I have to say I've sort of expanded this a bit. Is if you have a ONOO-Cycl disease, then of course the way to treat it is by downregulating the cycle biochemistry, and that is you want to treat the underlying cause. You don't want to just treat the symptoms, and so basically, what that predicts then is that things that lower different parts of the cycle should be helpful, and what it also predicts, and let me just say, so this is another way of looking at the causality of the cycle in the disease, right? But it turns out there's another way of doing this as well that tells you something about the causality of the cycle. And that is, if there are genetic predispositions that make people more susceptible, that raise cycle elements, that's a sort of an independent type of evidence for this fifth principle, okay? That the cycle is causal, and therefore what you need to do is lower the cycle, okay? So that's okay. So let's okay. So there are 34 different mechanisms that are currently this. Let me say the ONOO-Cycl tends to change from, over a period of 2 years. I make changes, and the current one has 34. Actually, there are 2 others that I put in there recently. 34 distinct mechanisms, and those mechanisms are all provided in this paper that I published on pulmonary hypertension. So this is another paper that makes a strong case, not as strong as the heart failure case, but still a very strong case that another disease, pulmonary hypertension, is a probable ONOO-Cycl disease. This is another paper that is publicly accessible, so anybody can get this paper for free and read it. And you just go into the ISRN hypertension site, and you can probably search under my name and you'll find it. And or you could put my name in pulmonary hypertension, you'll find it. So that's but basically what I did in that paper, I listed all 34 mechanisms and I documented each of the 34. So you can go and look at the literature that supports each of them, okay? Okay, so now, as I said before, the only thing that's truly novel about the cycle is when you put these mechanisms together, you get these cycles. And that's the only thing that I'm, sort of claiming is original here. But the consequences of this, as I think you can see, are quite stunning. Okay, I've got a list of these 34. We don't have time to talk about them. And you can look at them, as I say, in that ISRN hypertension paper. You can also look at them in the heart failure paper, but I don't document them in that paper, okay? So anyway, there's all these things that go on, and these are all, well-documented mechanisms. They're not things that I dreamed up after drinking too much on a Saturday night. So, okay, and now we're back to the 5 principles. So basically, we're going to go and look at these 5 principles now and look at how well they fit with a specific disease. And the disease I'm going to talk about first is heart failure. This is a table of, I believe, 19 different stressors that can initiate cases of heart failure. And so basically the question is, and you'll remember now, the first principle is that stressors in a ONOO-Cycl disease should work by raising cycle elements, right? And so what I did was I looked at the literature, and one of the things that's great about heart failure is almost everything in the world has been looked at. There's this incredible detail of information. And what you'll see there is that each of these initiating stressors raise cycle elements, at least 2, and in many cases more than 2, in the context of heart failure causation, okay? In that context, we know that these things act to raise cycle elements. So we have, there are 19 of these, and obviously I don't have time to talk about them, but what you can see is that each of them raises cycle elements. And so we have, lots of different things. Duchenne's muscular dystrophy, endotoxin, and exposure, and sepsis. This is a a cardiac-specific INOS overexpression. So, there's a lot of transgenic mouse models where you take, in this case, the INOS gene and you overexpress it in the heart only in a transgenic mouse. And what happens? You get heart failure, and you immediately get these direct effects on INOS itself, of course, nitric oxide, oxidative stress, mitochondrial dysfunction. All of those clearly occur in this mouse model. Anyway, lots of different things. Chagas disease, raising cytokines. There are lots of ways you can do this. So basically all of the, okay, so the first principle is clearly, strongly supported for heart failure, right? It turns out that 11 of the 12 elements of the cycle are clearly elevated in heart failure. And the only one where there's some question about it is intracellular calcium. But what's interesting is, although intracellular calcium itself may be somewhat in doubt, there's excessive activity of 3 calcium receptors: calpain, calcium calmodulin kinase 2, calcineurin, and each of those has roles in heart failure. So we know, in fact, that that even though the measurements of calcium can be interpreted in different ways, that calcium has an important causal role. Elevated calcium has an important causal role in heart failure, acting through these receptors. And all the other elements are clearly elevated, and all 12 of the elements then have causal roles in causing heart failure. Okay, so how can you do that if this is not an ONOO-Cycl disease? And clearly heart failure is a local disease, and you can show that in a number of ways. There are local changes in gene expression. You have these transgenic mice where you have only changed the heart and you get heart failure. You have a lot of changes in the, that can be detected by just simply examining the failing heart in animals or in humans after they've died. And you see local effects in the heart. And there are also different types of heart failure that can best be interpreted as being due to different tissue distribution within the myocardium. And so, for instance, you have right versus left heart failure. You have heart failure with and without arrhythmias. You have heart failure that shows valve dysfunction and other types that don't. So you have different tissue distribution. It doesn't have to be the whole heart muscle that's impacted. It can be parts of the heart, and you can get various things. So shows a local mechanism. Now, the other thing I did, and this was a huge table, looking at the activities of various cycle elements in producing the incredible complexity of the disease. Okay, so this is an absolutely stunningly complex thing. There are many changes in particular proteins. This is actually the calcium ATPase I mentioned. That's particularly important in the heart, and but there are changes in creatine kinase that inactivate it. There's changes in the electrical control of the heart. Changes in calcium cycling. You get changes in the control of the heart, and in many ways, you get 3-nitrotyrosine, which is a marker for peroxynitrite elevation. You have some of these other things that we talked about, NF-kappa B, TNF-alpha, one of the cytokines. So, you have many changes in mitochondrial energy metabolism. You have changes in gene expression, huge changes in gene expression, both within they were just looking at the proteins in the mitochondrion, but there are also other extra mitochondrial proteins that are changed. And what's true, and you get changes in the ryanodine receptor, you got changes in you get oxidation of cardiolipin. that's part of the this big cascade that impacts the mitochondrion. You get increases in matrix metalloproteinase activities. How does that work? It works simply because peroxynitrite and oxidants activate the precursors for those matrix metalloproteinases, and these then degrade a lot of the proteins in the failing heart and produce a lot of the changes that are seen. So you know, it's absolutely stunningly complex disease. Every single thing that I could find, you get fibrosis, can be tied back to activity of cycle elements. And so here were the ones we talked about before: peroxynitrite, superoxide just in the mitochondrion here, hydrogen peroxide derived from mitochondrial superoxide, oxidative stress. Anyway, it goes on NF-kappa B, cytokines, nitric oxide, etc. Mitochondrial dysfunction, calcium effects. What you see here is that every single cycle element has important roles in this process, all 12 of them, in producing specific changes that are characteristic of heart failure. You can't get that if this is some other disease. You know, if this is not an ONOO-Cycl disease, you're not going to see this. This is absolutely compelling evidence. So what I think is true is the ONOO-Cycl is no longer a theory. It is an established mechanism of disease. What it does then, of course, is it strengthens the argument that this is likely to be involved in a number of other diseases, depending on where it's localized. And as I said before, we have the paper on pulmonary hypertension when it's localized in the pulmonary arteries. It's the probable cause there. So there are a lot of different conventional diseases that can be explained by this. And you know, what I have to do, and what's difficult to do, is to take each disease and go through the kind of detail that I went through in heart failure and see if it's a good fit or not. And there's probably several lifetimes worth of work that needs to be done just there, okay? So anyway, okay, so what? Okay, so we're going to now talk about mainly MECFS and MCS. I'm not going to say much about these other two, but this is a table now. So I'm going to start talking about these as possible ONOO-Cycl diseases. And what this table does is it talks about various kinds of stressors that have been reported to initiate cases of these different diseases. And so with MECFS, you have a bunch of them. With MCS, you have a bunch. You have with fibromyalgia and so forth. Post-traumatic stress disorder, only 2 that I could find. And what I did was I used different colors to indicate how these can work on the cycle. Okay, so a number of these work on the inflammatory part of the cycle, and those include the infections, of course, but also include ionizing radiation, which actually works by raising NF-kappa B. Okay, and so those are the red ones. The blue and the green ones are actually somewhat related. They both involve excessive NMDA activity, but the green ones work directly on energy metabolism to produce excessive NMDA activity. And I'm going to say a couple of words about that in a little bit, but I want to say that what you have here, and so all of the MCS, all of these stressors that start MCS, as far as I can tell, work to raise the NMDA receptors. And the NMDA receptors have a key role in MCS. And that's, I think, a very important conclusion that needs to be, you know, that has very important implications, okay? Now, let me just say that this hasn't been done with ochratoxin, but it's been done with all these others. You can take each of these types of compounds, and you can treat animal models with them, and you can lower the toxicity of each of these by using an NMDA antagonist, okay, using a drug that lowers the NMDA receptor activities. And what that tells you is that the excessive NMDA activity is very important for the toxicity of each of these in the body, okay? Now, nobody's done that with ochratoxin A, but we know that ochratoxin A can raise glutamate, and glutamate is the main physiological agonist for the NMDA receptor. So one can predict that ochratoxin A does the same thing, but nobody's done the direct experiment, okay? And so that's, carbon monoxide is kind of interesting. I mean, you say carbon monoxide, okay, we know it poisons energy metabolism, right? And so you think then, well, we know everything we need to know about it. But the fact of the matter is, if you treat an animal with carbon monoxide and give that animal an NMDA antagonist, that animal will often survive a treatment that would kill an animal that doesn't have an NMDA antagonist. So the excessive NMDA activity is very important for the lethality of that compound, okay? And the same thing is true with a number of other things that work on energy metabolism, okay? So I think, the excessive NMDA activity is a very important thing in the body. And that's sort of another important take-home lesson here that I want you to get, okay? Now, there are a number of really interesting studies that have been published on CFS-ME. Well, actually, we're calling it MECFS now, so this is an old slide. And having to do with cortisol. So there's low levels of cortisol, and so that's important. And in fact, one of the genetic studies is not on cortisol, but it's on the receptor for cortisol. Okay, so you can lower the receptor, and that tends to increase susceptibility to the disease. So cortisol has an important role, and I'll talk about what the role may be later in my talk. You had this study by, I'm not sure I know how to pronounce this, Vladuciu. It sounds like a Romanian name, and Nadelson, who showed that angiotensin converting enzyme, when it's hyperactive, produces increased susceptibility. And we know that when the enzyme's hyperactive, you get more angiotensin II, and that increases the activity of this enzyme. And what does that enzyme do? It produces a lot of superoxide. Okay, so this is evidence that superoxide probably has a role in MECFS, and so that's sort of the key take-home lesson here. And it turns out that there's also genetic evidence in MCS that excessive superoxide has a role, again from genetics. There's evidence that excessive activity of some of the inflammatory cytokines tends to increase susceptibility. That's also true, by the way, for fibromyalgia. And they looked at different genes in the two systems. I don't know that it makes that much difference which genes you look at, but each of these cytokine genes, when they're hyperactive, tends to make people more susceptible to the disease. So you can see how these things then provide some evidence for the role of ONOO-Cycl elements. And there's Bowles et al. Studied people with maternally inherited mutations in the mitochondrial genome, And these produce very, very complex symptoms, but among those are MECFS. And so we think that, and he actually thinks that the main effect here is producing higher levels of superoxide rather than lowered ATP. And and I don't know that he's published that. That's what he told me several years ago. But so this has to do with the question that I sort of started out this talk regarding, and that is, how do you you want to test your theory based on evidence that wasn't in existence when you constructed the theory, right? And the ONOO-Cycl actually went through two major sort of forms. The first form that I proposed way back in January of 2000, and then more recently around 2007, where some other elements came into it, okay? And so in January of 2000, I proposed iNOS was involved, the cytokines were involved, NF-kappa B, oxidative stress, excessive calcium, peroxynitrite, mitochondrial dysfunction, superoxide, and nitric oxide. So all of those were proposed in January 2000. And there were just these three where we really had good evidence, okay? iNOS, cytokines, mitochondrial dysfunction. Those were clearly in evidence. And so then the question is, what about the ones where we didn't have evidence, where there were no's here? And it turns out that now, at this point, we have evidence for 6 of them. And the oxidative stress has been studied almost incredibly. There's somewhere, I think, like 26 different papers now that show oxidative stress in MECFS. So there's a huge amount of evidence there. There are 3 studies that show elevated levels of nitric oxide, and there was one that argued that there wasn't any. I'm probably a little bit prejudiced about that, but I think that one might be flawed. But anyway, but there are 3 anyway that showed elevated levels of nitric oxide. One of them was mine. So here you have 6 things that were predicted, and in each case the prediction was valid. In 2000, I added 3 more elements. 2007, added 3 more elements to the cycle, and the NMDA activity we already had evidence for. These 2 we didn't have evidence, but now we do with BH4. We don't have evidence on the TRP receptors here. So that's the only thing. But basically, what's true is that you can see there are 8 predictions where we had no evidence, and now 7 of them we do have evidence, and all 7 of them confirms the prediction. So that's pretty strong evidence that this is a ONOO-Cycl disease. And okay. So I think I'm going to skip through this except for one thing. This was a study that I published with 2 co-authors last year, and was a study of 17 MECFS patients. And the 3-nitrotyrosine, which is a marker for peroxynitrite, which hadn't been studied before in these patients, was extremely elevated versus healthy controls, okay? And what's equally, so we compared 17 MECFS patients with 111 healthy controls. The levels of these were, if you want to take a slide, you got to take the flash off. It doesn't work otherwise. The levels of these are completely non-overlapping, okay? So you've got your 111 healthy controls down here, and you've got your 17 MECFS patients up here, and there's a gap between them of a factor of about 2.25. So they're completely non-overlapping. This is the only thing I know of where that's true. And I think the fact that peroxynitrite is the central element in the cycle, and this is basically measuring, this is a measure of peroxynitrite, sort of tells you peroxynitrite is very important in this disease, and it clearly is highly elevated in these patients. Okay. Now, it was also true that BH4 levels were significantly lower than the healthy controls, and we found, and this was something I predicted actually, that sauna therapy works at least in part by raising BH4. Okay, and that's what I think is probably the primary mechanism of sauna therapy. The rate-limiting enzyme in the de novo pathway for BH4 is an enzyme called GTP cyclohydrolase 1. Okay, and its levels then regulates how much BH4 you get, and it turns out that that enzyme GTP cyclohydrolase 1 is stabilized by a heat shock protein known as HSP90. Okay, so HSP90 is a protein. Heat shock proteins in general, of course, go up under heating, right? Heat shock HSP90 requires actually only relatively modest heating to increase this, and when it does it, then it stabilizes the GTP cyclohydrolase 1, so you get more activity. And in fact, what Tapan Adya, who did these studies, he actually measured that enzyme and showed it was elevated by sauna therapy. So, there's a second mechanism, actually, I think, that's going on as well, but we don't have time to talk about all this stuff. So, so here's a situation where essentially what you're doing is you get a systemic rise in BH4 from the external parts of the body that are being heated, and those then, that BH4 can feed the other tissues. Okay, and so here's a situation where you're sort of treating the thing systemically, but it can have a favorable effect. And in fact, there is a literature that argues that sauna therapy can be helpful with MCS particularly, because it's been studied there most extensively, but also with fibromyalgia and with the MECFS patients. So basically what we're doing here is we're in this study, we were raising BH4, and that in turn actually did lower 3-nitrotyrosine, but not that much. I was hoping we'd see a bigger effect than we saw. You do see a lowering here, but it's not that high. So there's still a lot of oxidative chemistry going on, and so because of that, after you do your sauna therapy, and we measured these sort of 2 days afterwards, after you do your sauna therapy, a lot of the BH4 has already gotten oxidized later on because you've got all this oxidative chemistry going on. So you actually see bigger effects on the oxidation products than you see of the BH4 because of the way we did the experiment. I'm sure if we looked earlier, we'd see more BH4 effect. Okay. Now, I think I'm going to skip through this because we have a lot of things to talk about, but basically the question is, what's the central focus of MECFS? And what I'm going to do, basically, what I've argued in these slides, and I've made this argument before, is the probable central focus is in the thalamus, and that influences the neuroimmune system, and that there's a main effect on cortisol, and specifically cortisol in response to exercise. Okay, and there's some evidence for this that's discussed here. So and what I think is true, and again we don't really have time to go through all this, is that cortisol can act via a self-surface receptor to stimulate the calcium ATPase mechanism. Remember we talked about that, and lower intracellular calcium. And so I think what's happening here is that exercise, the stress of exercise, can actually raise intracellular calcium. And if you don't turn this cortisol on to lower it, what happens? Well, you get too much intracellular calcium, you get lots of nitric oxide. And in fact, that was shown by this Spanish group, Suarez et al. They showed that with MECFS patients, when they exercise, you get a huge increase in nitric oxide. This is sort of a whole body measure of nitric oxide, and it occurs within a few minutes. So this is very rapid. Okay, and so I think that's probably the way why MECFS patients react negatively to exercise. Why it's so traumatic to their biology, and so anyway, I think the only thing I want to say here is that we still have people who argue for so-called graded exercise as a misguided treatment for MECFS, and this is directly contrary to the literature that goes back all the way back into the to when CFS was first studied. You don't want to do that, and the best thing for people with MECFS to do is rest. And anyway, that's my opinion. Let me say I'm a PhD, not an MD, so none of this should be viewed as medical advice. Okay. Okay. Now, I don't know. Do we want to take a 5-minute break, or should we go on? What's your choice? Break. Let's take a 5-minute break, and then we'll come back. My next talk is on multiple chemical sensitivity, MCS, and one thing that's important about MCS, and I showed you this in the table before, but each of these 7 classes of chemicals acts to increase the NMDA receptor activity, and I've already said that. But so basically, what's true is that we have the pathways of action of 6 of them listed here. The 7th is carbon monoxide, which probably acts via energy metabolism, as I mentioned before. And so what we have here is 3 classes of pesticides: pyrethroids, organochlorine pesticides, organophosphorus, and carbamate pesticides. And they each act along different pathways to produce excessive activity of the NMDA receptors. Hydrogen sulfide acts itself along 2 pathways, one through these TRP receptors, and the other is actually directly on the NMDA receptors. So all these others act indirectly on it, okay? Hydrogen sulfide is the only one here that acts directly on it, although there are things that can inhibit glutamate transport, and glutamate is the classic, what's called an agonist for the NMDA receptors. So glutamate activates the NMDA receptors, and mercury acts to lower glutamate transport, and we know that. So we have this pathway, we have, now this is the big pathway, organic solvents, but it includes a whole bunch of things which are called sensory irritants. And they all act on these TRP receptors. 2 of them particularly, TRPV1, TRPA1, and probably the TRPA1 is the more important of the 2. And and they then lead to increased NMDA activity. You've got these, again, organic chlorines work by a GABA-A receptor. So they inhibit the GABA-A receptors, and they inhibit the NMDA receptors. So you have a double negative here, which gives you a positive, right? You actually have a double negative here that gives you a positive, and you have a double negative here. These little upside-down T's are each negatives and gives you a positive. So all these not only regulate the NMDA receptor activity, but they stimulate it, okay? All of these things, every single one of them. And so that's important. And okay, and so as I mentioned before, you can lower the toxicity of members of each of these classes by using an NMDA antagonist. So we know that not only it happens, but it's very important for the toxic responses in the body. Okay, now it turns out that there are 6 other kinds of evidence, and that each of which argues for the excessive NMDA activity in MCS, okay? And I think I'm just going to talk about 3 of them briefly. One is that this that the MCS patients are sensitive to monosodium glutamate, and so glutamate, of course, is the compound that stimulates these receptors. There's an allele of the CCKB receptor gene, which produces increased NMDA activity, and it's been shown now in 2 studies that when that allele is present, the person is more susceptible to MCS, so they have a bigger chance of coming down with MCS, okay? And there's a drug called dextromethorphan that's been used to treat MCS patients. And let me just say, I'm not saying that this is always a good treatment, but it is something that has been used in certain parts of the world. This is a over-the-counter drug that's used as a cough suppressant. That doesn't mean it's completely benign, and in fact, some of these patients don't tolerate it well, but it can be used. Anyway, and so there are a number of other types of evidence supporting excessive NMDA activity. So we think the NMDA receptors have an absolutely key role in MCS based on multiple types of evidence. And now, Iris Bell, who's a researcher at the University of Arizona, argued that one of the mechanisms in MCS was neurosensitization, that there's a change in the synapses in the brain, in regions of the brain which are impacted by these chemicals, and it makes those regions of the brain hypersensitive to stimulation, and in fact there was a study by Hillert who did brain scan study who showed that a particular region of the brain might be involved because it was hypersensitive to changes on chemical exposure, okay? And in MCS patients as compared with normals. So there are a number of things on this. But it's also true that you have sensitivities in MCS patients in other organs independently of any effect on the brain. So you get reactive airways disease, which is a form of chemically induced asthma. So you can get hypersensitivity in the lungs, you get rhinitis responses, you get sensitivity responses in the skin, in the GI tract, in the eyes, sometimes other tissues. So what I argue is that you have this local mechanism, and of course it involves the ONOO-Cycl, but also involves some of these other things. And that the tissues that are impacted in different MCS patients are not all the same. So you do see differences in symptomatic patterns, and so they're not all the same, and we shouldn't expect them to be the same, because you can get a number of tissues that can be independently become hyperactive. And now, William Meggs, who's an MD at one of the medical schools in the U.S., published 4 papers showing that when you have peripheral tissues, you can take a little tissue sample and study it under the microscope. And what he showed was that there were characteristic histological changes that were characteristic of neurogenic inflammation. And so now what is true is, and so he published these 4 papers on it, and I think that's an important observation. What's true is that sensory irritants, which are the common chemicals that MCS patients tend to react to every day, because there's so many of them, and those can act on these TRP receptors, which can in turn stimulate neurogenic inflammation, which in turn stimulates the NMDA receptors. Okay, so this is the sequence that's involved in a lot of these peripheral tissues, and it may also go on in the brain as well. But in the peripheral tissues, you can study it. You can't do a biopsy of the brain and see what's going on. Or at least I hope you can't. Now, Eva Milkvist, how do you pronounce her name? She's from Sweden. Huh? Milkvist? Okay. She's published close to a dozen studies now on the fact that many MCS patients are hypersensitive to capsaicin. So capsaicin is the hot stuff in hot peppers, okay? And capsaicin is the classic stimulator of these TRPV1 receptors, okay? So as I said before, we think these are hyperactive in MCS, and what she shows is that the MCS patients, many of them, are easily stimulated by levels of capsaicin that don't affect most normal people, and what you end up is with a cough, okay? So you can spray a little of this into the respiratory tract, and the person doesn't know whether there's capsaicin in there or not. But when there's a low level of capsaicin, if you have an MCS person, in most cases you get a cough response, whereas with most normal people with that low level, you don't get a cough response. So it's clear that people are hypersensitive. So this is a real direct measure that shows that these people are hypersensitive, okay? This isn't a psychological thing. It is a real hypersensitivity to something that we expect to be effective because it's working via one of the TRP receptors. And so the sequence of events in producing the cough response is this sequence where capsaicin stimulates this TRPV1 receptor, that stimulates neurogenic inflammation, stimulates the NMDA receptors, and you get a cough response. Now, the reason that dextromethorphan that I mentioned to you earlier is a cough suppressant, it's used as a cough medicine, is because it lowers the NMDA activity here and therefore can lower cough responses, okay? Okay, so what I'm trying to do is to get you to see how all these things really fit together very nicely. And so I reviewed her studies in my 2009 MCS toxicology review, and there's still further evidence that she's published, which is actually quite interesting since then. So anyway, but basically I think what's happening here, with the NMDA receptors is that, when you activate them, you get calcium, you get NOS, eNOS, you get nitric oxide, and you get peroxynitrite, and that, of course, reaches very deep into the ONOO-Cycle along this pathway, and so that's how we think that these various chemicals are acting to turn on the cycle by through that mechanism. Okay, and anyway, so there are a number of ONOO-Cycle elements that have been studied. Actually, there have been more studies done on animal models than in humans. And we've got, NMDA activity, we've got nitric oxide, we've got peroxynitrite, we've got oxidative stress, we've got inflammatory cytokines, we got calcium. But you also have things like neural sensitization. You can show changes in brain sensitivity in these animal models. And so a lot of these things can be have been studied in animal models. There are over 40 papers on this that have showed this, and they showed that you do get real sensitivities, and you can get cross sensitivity. So you initiate sensitivity to one chemical, and then you can look at another one and have sensitivity to that as well. Okay, so a lot of the basic properties of MCS that we see in humans can be replicated in animal models, and other changes that have been reported in animal models include neural sensitization, neurogenic inflammation, airway sensitivity, so the changes in the lungs, inflammation. Okay, so you can see changes in inflammatory cytokines. You get a breakdown of the blood-brain barrier in animal models and in humans with MCS. Okay. And I can tell you how that works, but I don't think I'm going to spend the time on it right now. So, and we know it can be produced by peroxynitrite and downstream effects of peroxynitrite. So you get a breakdown of the blood-brain barrier. And so now, the most important studies that have been done on MCS are the genetic polymorphism studies of genes that are involved in the metabolism of chemicals implicated in MCS. And the reason that they're important is because they provide direct evidence that chemicals cause this disease, okay? Okay, so we're going to talk about genetic polymorphism studies, and then we're also going to talk about other genetic studies that don't have to do with metabolism of chemicals, but have to do with other things. And so this is a table that that summarizes these genetic polymorphism studies, and that involve, what is it, 7 genes, I think, on 1, 2, 3, 4, yeah, 7 genes, okay? And so all of these appear to have roles in determining susceptibility to MCS, and all of these have roles in metabolizing chemicals that we think are involved in causing MCS, okay? And in fact, what these studies do then is to give you compelling evidence that these chemicals, in fact, are causing the disease, and therefore the metabolism of those chemicals can influence whether you get the disease or not. Okay, and so what are these? What are these? This PON1 is an enzyme which is involved in the detoxification of organophosphorus toxicants. Okay, and those involve both organophosphorus pesticides, which are a very important group of chemicals that have been involved in causing MCS. I suspect they're less involved now because that particular group of pesticides is much less used now. There's a cytochrome P450, CYP2D6, which was studied by McKee and Eisen and her colleagues, and also by Berg, and I'll say something about that. Berg's a group here in Denmark. And what does CYP2D6 do? One of the things it does is it hydroxylates these same organophosphorus pesticides, okay? So these organophosphorus pesticides are typically used in an inactive form. And both our bodies and also the bodies of insects have an enzyme, in our bodies it's CYP2D6, that can convert them into the active form, which is toxic to both insects and humans, okay? And so this enzyme then is involved in activating those pesticides. It's also involved in activating some other compounds, some hydrophobic compounds, which then become more active on these TRP receptors, okay? So it can have both of those effects. So the prediction is that higher activity will produce increased MCS, and that's what the genetics says, okay? The form of the enzyme that gives you higher activity gives you greater susceptibility. So you tend to have that more commonly in people who have MCS, okay? On the other hand, there are a whole bunch of other things, these last four, that are detoxification processes. So the more activity you've got there, the less likely you're going to have MCS, and that's exactly what's been shown in these studies, okay? And there's another enzyme, NAT2, that's involved in acetylation. This is more complicated because acetylation of some compounds can activate them, acetylation of other compounds can inactivate them. So you can't make a simple prediction without knowing what compounds people are exposed to, and in most cases we don't. So, but it was shown that this does affect, in 3 different studies, this was shown to influence whether people came down with MCS or not. And if I remember, one of those studies, it seemed like more activity was involved, and in another study, it seemed like less activity was involved. So different populations of people are exposed to different chemicals. You cannot assume the same pattern for these genetic effects when you have different populations exposed to different chemicals. They're not necessarily going to be the same, okay? And so that's a very important thing. And so one of the things I want to point out here is that these last 4 studies that were all done by Schnackenberg and his colleagues in Germany have extremely high levels of statistical significance, okay? So less than 1 chance in 10,000, less than 1 chance in 10,000, less than 1 chance in 10,000, less than 1 chance in 1,000, okay? These are not statistical flukes. You know, these are obviously real effects. I mean, the statistical significance tells you that these are extremely powerful experiments. And not coincidentally, this was the largest study that was done. Now, that's what you expect when you have a real effect, right? The more people you study, the higher the level of statistical significance you're going to see. And so they showed extremely high levels of statistical significance here. And on these 4 genes. So what we have here is different studies by different people, but all basically showing very much the same kind of pattern. And all of these then argue very strongly that MCS is a disease of toxic chemical exposure, because that's the only way in which these genes that influence the metabolism of these chemicals can influence whether you get MCS or not, okay? That's something very important, okay? And it has to be repeated over and over and over again. The notion that this is not a response to toxic chemicals is sheer nonsense based on the genetics, okay? Now, there's only one study that I know of that shows a different, a result that's different, okay? And that was the study of De Luca in Italy, where she and her colleagues looked at CYP2D6 and didn't see any effect. So CYP2D6 was shown to have a role by McKee and Eisen and her colleagues in Canada. It also appears to have a role from the Berg studies that were done here in Denmark, but it was a non-significant trend. But it was a smaller study. So when you do a smaller study, it's going to be harder to reach statistical significance. In fact, the pattern that Berg showed was exactly the same as the pattern that McKee and Eisen showed, except that McKee and Eisen had a bigger population, and it reached statistical significance. In Berg, it didn't. It was a non-significant trend. So, in fact, it tends to confirm the McKee and Eisen study. Now, why is the DeLuca study different? Well, I don't know, really. It could be that the chemical exposures in Italy were different, and that may be the simple explanation. That's quite possible. Or it could be it's just a statistical fluke, and if you went and did it again, you'd see a statistical significance. We don't know. She also looked at a bunch of other genes that nobody else has shown to have a role, and she didn't find they had a role, but that's not any kind of conflict. So basically, what we see here is a high level of confirmation of this pattern, and I think it's important. And as I said before, particularly the Schnackenberg studies are very convincing because of the extremely high level of statistical significance. Okay, so I think there can be no question from the data that MCS is a disease of toxic exposure. And now what's also true is that there were also some other studies. Now, these are not studies done on genes that are involved in chemical metabolism, but in genes that are involved in things that we think are linked to the ONOO-Cycl, okay? And one of them I mentioned before was the CCKB receptor gene. And what was shown by Binkley et al. In 2001 and by Berg et al. Here is that a particular form of that gene, which produces higher levels of NMDA receptor stimulation, was associated with the MCS patient. So apparently that causes increased susceptibility, and that then gives you then another type of evidence here, genetic evidence, that the NMDA receptors have roles because they're regulated by this CCKB receptor. There was another study that came out of Japan. Actually, I think I have the wrong thing cited here, but anyway, it's the same group. And what they showed was that SOD2, this is a superoxide dismutase in the mitochondrion, which we think is particularly important, that form polymorphism of that's somewhat less active is associated again with MCS. Okay, and so again that provides evidence that superoxide has a role. In this case, superoxide in the mitochondrion has a role in So the superoxide gets rid of superoxide dismutase, which is produced by this gene, gets rid of superoxide, and so what you find is you have lower activity in this form that's associated with MCS, and that means the superoxide levels are higher. Okay, so all. When you look at these things, they all make sense, not just as things that you see statistical significance, but in terms of the direction of the effect, where you can make a prediction on the direction of the effect. That's what you see from the evidence, okay? And I think that's, again, it's very important. It's very important not just to look at these as numbers, but as what is the mechanism that's going on, and how does that relate to what we think is going on in MCS? Does this confirm what we think is going on? The answer is yes, it clearly does. And I can even go back and let me go back to this. This is kind of interesting because the PON1 polymorphisms differ not so much in terms of activity in general, but in terms of activity on specific compounds. Okay, so you have different specificities of the enzymes, that some of them are more active on certain compounds and others are more active on other compounds. Well, why am I telling you this? Because the Haley study, that H there, they studied the Gulf War syndrome people. We think Gulf War syndrome people were exposed to sarin gas, right? From Saddam Hussein's rockets that were coming in that had sarin gas. And whereas, the McKee and Eisen studied civilian Canadians. What are they exposed to? Well, they're probably exposed to pesticides. Okay? So it turns out that that the form of the PON1 that is less active in metabolizing sarin gas was found more commonly in the Gulf War syndrome vets, the people who came down with Gulf War syndrome. They're less able to metabolize the toxicant. McKee and Eisen showed a different form was associated with the civilian MCS patients in Canada. That makes sense too, because they're exposed to pesticides, and you have a different form of the enzyme that's less able to detoxify the organophosphorus pesticides. So all these things make sense when you look at them. They go exactly the way you predict, and so okay. So basically, what this says, basically, I think, is there's no question that this is a disease of toxic exposure. Now, what I want to do is to talk about a paper here that comes to the opposite conclusion. Okay, and this is the Hetherington and Battersill paper that was published in January of last year in this journal, and it was entitled "Review of Evidence for a Toxicological Mechanism of Idiopathic Environmental Intolerance." And so they're calling what I've been calling MCS, they're calling it idiopathic environmental intolerance. That's what the industry people like to call it. Or IEI, if you will. And so, I saw this and I said, oh, this is going to be an interesting paper, right? We're going to see them go through each of these studies that I've just been talking about, right? Okay, so let's see what they. So one of the things that was one of the things they state in the first paragraph of the introduction is there's no internationally accepted diagnostic criteria for IEI, and it's very difficult to assess the reliability of published diagnoses for IEI. That's true. With MCS, it's absolutely not true, because the 1999 consensus case definition, which was published by, if I remember correctly, 34 different prominent scientists who signed it, provided a set of criteria for diagnosis. Okay, IEI, you don't have it, so why should you call this IEI? It's totally irrational, right? But that's what they're doing. Okay, so let's. So let me just say, the paper that I published in 2009, the MCS Toxicology Review, is a paper that I am very proud of. I was chosen from all people in the world to write that paper by the 3 editors of this publication. Okay, and And it is the only paper that has been published in a well-recognized toxicology publication that basically, describes and gives toxicological evidence for the, for MCS as a disease of toxic exposure. And let me just say, obviously these editors would not have asked for a paper to be written if they were not already convinced that there was good evidence that MCS is a disease of toxic exposure, right? Why would they ask for somebody to write a paper on it if they didn't think that it was a toxicological issue? So all of those things, I think, clearly make this paper an extremely important paper in this whole issue of whether MCS is a disease of toxic exposure. And what was interesting was that Hetherington and her colleague, Battershill, first of all, they don't cite that paper. They don't cite either my 2002 FASIP Journal paper or my 2007 book, which were precursors of it, which also had substantial evidence on these things. And yet they published a review on evidence for toxicological mechanism by failing to cite what are clearly the most important papers. So that's a little strange. Okay, let's go on. Now there are a whole bunch of false statements that they make in here, and there are also a whole bunch of things that are simply left out. So, for instance, the only thing when they talk about the sensory irritation and the idea that these irritants can feed in to neurosensitization and through the NMDA receptors, the only thing they have to say about it is this thing about neurotoxic effects produced by GABA A antagonists, and they quote this from the McKee and Eisen paper, time-dependent sensitization. But then they completely fail to point out that these are all mechanistically linked together. So they talk about sensory irritation. They talk about neurotoxic effects produced by GABA A antagonists. They talk about this neurosensitization process, and they talk about increased NMDA activity, but they don't link them together. They talk about them as being independent, separate things that are kind of sitting out there in space, not linked together, okay? Is that deliberate obfuscation? I guess I'll leave it to you to make that decision. Now, we know that excessive NMDA activity has a central role in generating what's called long-term potentiation and therefore neurosensitization. I haven't talked to you about this before. With both of them acting directly through increased intracellular calcium, nitric oxide, and other downstream effects, okay? So when you look, and this was actually the focus of my 2002 paper that I mentioned a while ago, was how these things are all kind of linked together, okay? So we know that these things are all linked together, and that that the for instance, the GABA A receptors can feed in through this pathway, but they don't link anything. So they don't link anything up. They just have these things kind of sitting out here in. Now, they make a statement here about Meg's work. It says, Meg's proposed that the term, quote, neurogenic inflammation in relation to IEI, referring to chemically mediated activation of C fibers, leading to release of vasodilator neuropeptides. Okay. They claim that Meggs proposed this term. Is that true? No, in fact, it's not true. Meggs' first paper on this was the 255th paper in the PubMed database on neurogenic inflammation. This was already a well-established, well-documented phenomenon when he first started looking at it. And so why they claim that he proposed this term when it takes about 20 seconds to show that that's not true, that's very disturbing, okay? And let me just say, and I'll say something about this a little bit later, but you know, we scientists, we do occasionally make mistakes. We're all human. But we also try to avoid making obvious mistakes to preserve our professional reputations. It's very important. And yet there are all these errors in this paper that are easily checkable. They criticize Milquist's study on capsaicin, stating there are no controlled blinded studies to show this is the TRPV1 response in the absence of knowledge of exposure. That's just simply untrue. You can do studies. The person doesn't know what's being sprayed, and you can show effects when there's capsaicin and not when there's not capsaicin. So you can do controlled studies. That's not true. You talk about my work. The author proposed the chemical exposures that might induce this mechanism could involve organophosphates, carbamates, and solvents. However, there's no direct evidence for this. And then they say it's difficult to conceive how the very wide range of chemicals cited triggered for IEI could all lead to elevated nitric oxide. Well, we've already shown you all of that. It's not difficult to conceive it at all. But basically what they're saying is there's no evidence that organophosphates, carbamates, and organic solvents can cause MCS or IEI. That's not true. They're also saying there's no evidence that these chemicals can elevate NMDA receptor activity. That's not true. And then again, they're saying that there's no evidence that increased NMDA activity can produce increased nitric oxide. That's not true either. So, you know, I guess I better, let me just say, in the genetic studies, this is the key part of their paper, is a statement. They make a whole bunch of statements in there. And each of them is either, first of all, I mean, this is the most striking thing, I think, is they talk about these genetic studies on these genes that are involved in the metabolism of these chemicals. And that linkage to metabolism is made absolutely clear in both the McKee and Eisen paper and in the Schnackenberg papers. You can't miss it. But they don't say why these studies were done. The reader has absolutely no idea what the significance of these studies are from reading this paper. So, you know, and then they make quite a number of things that they left out. For instance, the McKee and Eisen study, they talk about NAT2 and CYP2D6, but not PON1. They don't talk about the Haley study on PON1. They don't cite the Haley study. They go to discuss the Berg study that was done here, falsely stating that neither NAT2 nor CCKB showed any apparent association. That's false. Both of those do in the Berg study. They also failed to discuss the non-significant trend of the data for CYP2D6, a trend which is discussed in the Berg et al. Article. You know, if there's a surprise here at all, the Berg study was a pretty small study. the surprise is that the NAT2 data is statistically significant, actually. I mean, the surprise in that study is that they did show statistical significance even in a relatively small study. That's the surprise, not the other way around. Anyway, okay, so basically what they do is they leave out everything that's important. They leave out the fact that the Schnackenberg study showed this extremely high level of statistical significance. They leave out the fact that these genes are involved in the metabolism of chemicals. They leave out, they basically dismiss it in one sentence. So when I went through this whole thing, this genetic study, well, let me go back. What was true was that they had 17, and this is just in the genetics section, this very short section of the paper. They had 17 errors, either important omissions or misstatements of critically important issues in the genetics, each of which appears to be designed to buttress their central false claim that IEI is not a disease of chemical exposure. And let me just say, the chances of having 17 random errors, if they were indeed random, and they would all be on one side of the argument and not on the other side, the chances of that happening are less, by just chance, by random errors, is less than 1 in 128,000. Okay. So that tells you something, right? Let me just say, so why did they publish this paper? I mean, it's full of omissions, misstatements, and outright falsehoods. I don't know the answer why they published this paper. What I'm going to do is to suggest to you that similar papers have been published in the U.S., and I think it's pretty clear why those papers were published. I'm not going to say that this is the reason they published their paper. I don't know why they published their paper. What's true in the U.S. is it's easy to publish a paper denying the reality of these diseases or the reality of their causation by particular environmental stressors. Having done that, and there are a number of such papers that have been published, the authors of those papers can hire themselves out as expert witnesses in trials defending insurance companies from lawsuits filed by people who suffer from these diseases. It's possible for these expert witnesses to charge $400, $500, $600, $700, even more per hour. And so if you have 20, 30, 40 hours on a lawsuit, you can make a lot of money. Okay. And you can defend the insurance companies. And it's possible for people over a period of 3 or 4 years in many lawsuits to make the financial equivalent of an unshared Nobel Prize with not much work. I mean, it's not that hard to do, okay? So, there is, those things do happen. Again, I'm not saying that this is why Hetherington and Battershill published this paper. I don't know why they published the paper. But these things happen. And now, what happens then is that, who loses from these lawsuits? Obviously, the people who are suffering from these diseases. Who gains? Well, the insurance companies gain because they don't have to pay out. And of course, what happens is if the insurance companies win a certain number of cases, then other people are less likely to file because they figure it's hopeless, right? So, so that's what goes on. Okay. The electromagnetic hypersensitivity has been a great puzzle. And one of the reasons it's been a great puzzle is there's been a great puzzle on how electromagnetic fields can influence our biology. And particularly electromagnetic fields for microwaves and other type of exposures where the individual photons that make up these fields are low energy. They don't have enough energy on the individual photons to influence the chemistry of our bodies. So if the individual photons can't influence the chemistry of our bodies, how can the EMF fields do it? And that's been a big issue. Okay, so I solved this puzzle in a paper that I published last year on EMFs that showed that these EMFs, and there are a variety of types of EMFs that did this, but I'm going to focus mainly on microwaves because those are the ones that are most our most concern. But there are also other types of low energy EMFs that do this. They act to activate what are called voltage gated calcium channels. Okay, so these are channels in the membrane, in the plasma membrane of cells that that basically when they're activated, they open up, they allow calcium to flow into the cell. It's the excess calcium in the cell that's responsible for most, if not all, of the downstream effects. Okay, so anyway, this is some of the data, and I'm not going to go through it. But basically, I think what's probably true is these voltage-gated calcium channels are also probably part of the ONOO-Cycl, acting more or less in parallel with the NMDA receptors, because when you have energy metabolism dysfunction, of course, that's one way in which the membrane can get depolarized, and it can activate these channels and produce increases in intracellular calcium. So the downstream effects of these channels and the NMDA receptors are going to be very similar. So basically what's true is that there are a whole series of similarities between the NMDA receptors and these, particularly the L-type voltage-gated calcium channels. These are channels that stay open fairly long time when you activate them, and therefore can produce a lot of calcium in the cell. And so they have a lot of similarities here. They both, when they're activated, they open up ion channels. They're both activated to some extent by partial depolarization of the plasma membrane. Both the ion channels stay open relatively long time periods. Both of them can allow a lot of calcium to flow into the cell, and the effects of both are mediated to a great extent by this elevation of intracellular calcium. Both can produce big increases in nitric oxide and peroxynitrite, and both can stimulate long-term potentiation and therefore neural sensitization. And this is known, this is empirical. This isn't just a prediction. And both are expected to initiate and stimulate the ONOO-Cycl. So basically, I think what's happening here is that EHS, the mechanism of EHS, is very similar to the mechanism of MCS. And the difference is that you have, with EHS, you have these voltage-gated calcium channels involved, and with MCS, you've got the NMDA receptors involved. They produce very similar kinds of responses. So the types of mechanisms that are involved in producing this high-level sensitivity in chemicals in response to NMDA receptors are probably very similar to the types of mechanisms that are involved in EHS. And so that's a very plausible argument. It's not proof, but I think it's highly unlikely that these targets would be so similar unless there was a real important mechanism here. Okay, so are you up for some therapy? Okay, so basically, let me just remind you again, the goal of therapy is to lower the cycle, to lower all of this stuff to the extent you can, okay? And so there are a number of ways of doing that, but it's going to be a big challenge because of the ways in which all of these different cycles sort of amplify each other, and that's the critical. Okay, so let's talk about some specific things. And let me say I could go on all night on these, but I'm not going to. one of the agents that is a powerful proxy nitrite scavenger is 5-methyl tetrahydrofolate. Okay, and that was shown both in this this res paper and also in this Antoniadis paper, and also in some other papers. Okay, this is a powerful scavenger of proxy nitrite. Okay, now what do people mostly think about when they see 5-methyl tetrahydrofolate? Well, cancer. Well, but what's the mechanism? These are methylating agents, right? This is the primary methyl donor in the methylation cycle. And so if you talk to 100 scientists about this who know anything about it, and you ask, what does 5-methyl tetrahydrofolate do? They'll say, oh, it's involved in methylation, right? The problem is it's also a powerful proxy nitrite scavenger. So when you raise 5-methyl tetrahydrofolate, it could be acting to stimulate methylation, or it could be acting to lower proxy nitrite, and you don't really have the foggiest idea which of those two it might be, at least based on first principles. Okay, so what I'm saying here is that the assumption in the literature is it's working to stimulate methylation, but it may not be. And I think that, anyway, people have used 5-methyl tetrahydrofolate to treat patients with these diseases, and it can be helpful. It's also true that they don't tolerate high doses, and how high a dose is too high varies with the patient. Okay, they're not all the same. Why should they not tolerate it? After all, you'd like to get rid of the proxy nitrite, right? It's I think what's happening is that when 5-methyl tetrahydrofolate reacts with proxy nitrite, it gets oxidized to 5-methyl dihydrofolate, and this thing breaks down to products which are lost to the folate pool. Okay, and I think at least one of those products, either the dihydrofolate or some of the breakdown products, must be toxic. So you get too much of this in there, you got a lot of proxy nitrite, you might generate some toxic products. That can be problematic. Okay, so this is a very common problem with these diseases. Is you have things that can produce certain favorable things, but there are also some problematic patterns to it. And let me just say again, I'm a PhD, not an MD. None of this is medical advice. Okay, I'm just trying to give you a feeling for what might be a useful way of looking at these things. Okay, so what? How can this be effective and not lead to too much of these oxidation products that may be toxic? I think one way of doing it is to use high doses of ascorbate, vitamin C, along with the 5-methyl tetrahydrofolate. Okay, why? Well, okay, first of all, ascorbate itself is a proxy nitrite scavenger. So by lowering proxy nitrite, it will also be helpful in that process directly. But the other thing is, ascorbate, of course, is a reducing agent, right? And one thing it can do in high doses is it can reduce 5-methyl dihydrofolate back to 5-methyl tetrahydrofolate. Okay, convert this guy back to that one, in which case shouldn't be a problem. No toxic product. Okay, so to some extent, you would expect then high doses of ascorbate to work in two ways to lower the toxicity and allow you to use higher doses of 5-methyl tetrahydrofolate. Okay, so how How much ascorbate should you use? Well, orally, typically people tolerate something in this range up to 2.5 grams. So depending on the person, how much they tolerate, you go up to whatever level seems to be well tolerated. And that might be something useful. A lot of people with these diseases have inflammation in the GI tract. And so when you're taking these orally, what's going to happen? You hit the inflammation in the GI tract, you hit a bunch of peroxynitrite there. And so if the stuff is oxidized, you have a dual problem. One is that it may never get to the tissues where it's most important to get to. And so you can see, but if you take oral ascorbate, all of that should be helped at least. So there are ways of it seems to me, of taking these understandings and try to use them to optimize therapies, to so that you can kind of get a feel for how you go about doing these things. And now, high-dose ascorbate actually can be viewed as a therapeutic agent in different contexts, and that is it can also act to help lower not only peroxynitrite but also to raise BH4. How does it do that? Well, it turns out the first oxidation product of BH4 is BH3, and it can be also reduced back to BH4 by ascorbate, so that's useful. Maybe I should I go back to that slide? I think maybe it would be useful to go back to that slide. This one here. So when peroxynitrite oxidizes BH4, the first oxidation product is BH3. BH3 is unstable. It typically oxidizes further to BH2 and other oxidation products. But BH3 can be reduced back to BH4 by ascorbate. So if you have high enough ascorbate, you can recover a lot of the BH3 and put it back here and lower this part of the cycle. So that's some people have used high doses of have used IV ascorbate to treat some of these diseases at much higher levels than you can get with oral. Something typically often on the order of 10 to 30 grams of IV ascorbate, and that seems to be helpful according to reports. So that's another thing that might be considering. I don't know what the legalities are of doing these things here in Denmark but obviously you have to look into those things. There are other things that can be useful here. I think that hydrogen peroxide, which is, it turns out that hydrogen peroxide is produced from high-dose ascorbate when you use it IV. Okay, you have enough. So ascorbate is a reducing agent. One of the things it can reduce at these very, very high concentrations is molecular oxygen. And so when it reduces molecular oxygen, you get hydrogen peroxide. And it's thought actually hydrogen peroxide can be useful as an anti-cancer, as a treatment for cancer. And people have used high-dose IV ascorbate in that way. That's even higher doses than the ones that I was just talking about. So anyway, but hydrogen peroxide also can induce NRF2, which is an important antioxidant regulatory factor, which I've gotten very much interested in, and I think it's a very important response system, but I don't have any slides in this set to talk about those. Anyway, yeah, okay, I guess I do have a little bit on it. Okay, so NRF2 can be induced by phenolic antioxidants, by including these that I've listed here, curcumin. It can be induced by gamma tocopherol and delta tocopherol, and also the tocotrienols, gamma and delta. So these are other forms of vitamin E that can do this. Alpha tocopherol, which is the standard form, does not do it. In fact, alpha tocopherol is counterproductive. Why? Because when you have high levels of alpha tocopherol, it causes the other forms of vitamin E to be degraded, to be lost. Okay? So you end up with deficiencies in the other forms, which are doing some really good things. Okay? Let me just say, my opinion is that the reason the vitamin E trials have failed repeatedly is because they're using the wrong forms of vitamin E at the wrong doses. Anyway, anyway, so there's a lot of other things that actually activate NRF2, and NRF2 is probably an extremely important thing to activate in terms of treatment of many diseases, many chronic inflammatory diseases. Okay, so let's go on here. Okay, I talked about sauna therapy before. And sauna therapy acts by raising the rate-limiting enzyme BH4 synthesis. Sauna therapy historically has been used by people who've claimed that it's working by a detoxification. Now, I'm not saying that it doesn't have anything to do with detoxification, but I think that what's probably at least as important is the fact that it can work in other ways, including raising BH4 levels. So I'm not convinced that the detoxification is certainly not the sole mechanism, and I'm not even convinced it's the main mechanism of action of sauna therapy or other kinds of heat therapies. And heat therapies, as I'm sure you know, are traditional things in many cultures. From Finnish baths and Finnish saunas and Turkish baths and Native American sweat lodges, and the Russians have them, and it's a common thing in many different cultures as an important therapeutic approach. Of course, modern medicine thinks it knows better and doesn't use these things. But so anyway, okay. Let me talk about a couple other things. Resveratrol is another phenolic compound that is thought to act mainly by raising the level of SIRT1, which in turn produces quite a number of protective effects. Lowers superoxide by at least 5 mechanisms, and it improves mitochondrial function. It lowers NF-kappa B activity, lowers INOS. It increases BH4 production. So essentially SIRT1, at least in principle, can lower the whole ONOO-Cycl, and it's really quite striking. So I think this is another thing that may be useful. But basically, I think one of the problems in terms of using it in these diseases is that there is a deficiency in NAD that's produced by the cycle. And I haven't talked about that part of the cycle, but there is a deficiency. And part of the problem is that the SIRT1 that I talked about here is an NAD-dependent enzyme. So you need to have normal levels of NAD for it to be active. So it may be important when you're using resveratrol to simultaneously use to use some nicotinic acid in order to restore the levels of NAD in the body. And what I propose, and let me just say, you don't want to use nicotinamide. Have I lost everybody? No. Okay. Nicotinamide is another form of niacin. Okay. So you got nicotinic acid, nicotinamide. Nicotinamide is itself an inhibitor of SIRT1. So if you use nicotinamide, you basically block SIRT1, which is, of course, you're trying to do the opposite here, right? So what you want to do is use nicotinic acid. I think one of the things that's useful is to use the no flush, low flush nicotinic acid, which is an ester between nicotinic acid and inositol, and which is hydrolyzed over time, so it releases the nicotinic acid over a longer time period and prevents flushing. So that can be a useful way of providing a lot of nicotinic acid and not having any unpleasant side effects. So anyway, those are some things. Magnesium there's a huge number of people in many Western countries that are low in magnesium for several reasons. One is we eat a lot of animal foods, which tend to be low in magnesium. We eat a lot of highly processed foods, which are low in magnesium. And with intensive agriculture, we've depleted magnesium in the soil, so a lot of our foods don't have as much magnesium as they should have in them. And so, for all of those reasons, we have an extraordinarily high level of at least marginal magnesium deficiencies, and often worse than that. What does the magnesium deficiency do? Well, one of the things it does is it gives you excessive NMDA activity, okay? And so that's a big problem. Now, why do I say that? Now, the NMDA receptors, they're receptors in the plasma membrane, and as I say, they can open up under some conditions and allow calcium to flow in the cell, right? It turns out that right on the center of the channel that could open before it opens is a magnesium ion. And so the magnesium ion has to come off before the NMDA receptors can be activated. But if you're low in magnesium, you probably have a bunch that are already, off, and so those things can get activated much more easily. And so then you get excessive activity of the NMDA receptors. I think probably all of us should be taking magnesium, because even those of us who have pretty good diets may be magnesium deficient. And the form I like is magnesium malate, although there are a number of other forms that are good, but I would avoid magnesium aspartate, because aspartate, like glutamate, can stimulate the NMDA receptors. So you don't want to do that. You don't want to use magnesium aspartate, okay? Which is another common form that is sold. So omega-3 fatty acids are important in a lot of things. They have anti-inflammatory activity. I found out the other day that they also activate the NRF2 receptors. That's really, or NRF2 protein, I guess I should say. It's not a receptor. And they actually also have a nice property for people who are electromagnetic sensitive, because they lower the sensitivity of the L-type voltage-gated calcium channels to activation. So they may be useful in that way as well. So there are a lot of things in which the fish oils, the DHA and EPA, the long-chain omega-3 fatty acids, may be quite useful. One of the things that happens in the process that produces mitochondrial deficiency in these diseases is you get oxidation of cardiolipin in the inner membrane of the mitochondrion. And when cardiolipin oxidizes, it greatly increases the amount of superoxide that's generated. So you already have a problem with too much superoxide, and that causes the oxidation, the cardiolipin, which then produces a much, much greater problem with superoxide. And so that, in fact, that's one of the things actually is very well documented that goes on in heart failure. A lot of these mitochondrial things that I haven't really talked to you about are extremely well documented in heart failure. And so in ONOO-Cycl disease, we know that a lot of these things are going on. How do you restore cardiolipin in the inner membrane? You can do it with phospholipids of various sorts. You have to be careful that the phospholipids not be oxidized, because sometimes they can get oxidized in the process of preparing them. But I believe that it may well be the case phosphatidylserine has a particular role here. Why do I say that? Because phosphatidylserine, it turns out, has a specific transporter that will take it right into the inner mitochondrial membrane, which is where the cardiolipin has to be made. Okay, so it may serve as a precursor. I don't know that, but it's plausible that that's what's going on. It's also true that carnitine and acetylcarnitine can help with mitochondrial function, and it also may have a role in regenerating the cardiolipin. And I guess I'm not going to tell you about that because we're running out of time here. So anyway, there are a number of these agents that have multiple effects, and you kind of have to get into some of the details to see how they can work, and they're not always simple. So, for instance, carnitine and acetylcarnitine. Acetylcarnitine seems to be more active than carnitine. Has been shown to affect one of the other glutamate receptors. And when it does that, it can actually lower the responsiveness of the NMDA receptors. So here's a totally different mechanism that may be helpful, okay? And so yes, acetylcarnitine and carnitine do help restore mitochondrial function, but that may not be the only thing they do. Is there a downside? Well, yeah, there actually might be, because it turns out that you can get from these, you can also get an increase in NF-kappa B activity, which of course you don't want. So, you know, these things are not always unmixed blessings, but basically I think there's a lot of empirical work that kind of goes into treatment of individual patients. And of course, individuals are also genetically heterogeneous, and they're also heterogeneous in other ways, which makes it a big challenge. So, you know, I think, you know, medicine often thinks, oh, you know, that patients are pretty much the same if they have a particular disease, but in fact, it's very clear that that's not true. And it seems to be particularly problematic with this group of people, because you have such a complex mechanism. There's so many different genes that kind of come in and influence it in various ways. Carotenoids act as a scavenger of the acid form of peroxynitrite, and so they can be useful. And the one that seems to be the most active is lycopene, one that comes from tomatoes. And so that's useful. Carotenoids also can activate NRF2. I haven't told you why NRF2 is so important. It is very important, but it's activated by a lot of things, a lot of useful products. So there are a lot of agents that lower NF-kappa B, including some of the phenolic and thiol antioxidants. And so that's another thing that can be a focus for therapy. So I think maybe I should quit at this point, and thank you for coming.
Thank you.
Okay.
Thank you for providing us the power of your spoken words to supplement our understanding. I think it has been very useful for me at least, and I think for others. Thank you.
Right.